capability for genome sequence-based infection control and MRSA example by Dr. Nigel Saunders. He's a medical doctor, originally graduated from Aberdeen. Then he got his PhD in Imperial College for medical bacteriology. He was the first recipient of Wellcome Trust Fellowship in Medical Microbiology in Oxford University. For 11 years, he was head of bacterial pathogen function genomic group. And last 5 years, his main focus is developing concepts and methods for new bacterial bioinformatics to make better use of pathogen sequencing for health and infection control. And in 2020, he became Chief Scientific Officer and co-founder of Genpax to develop the idea. Welcome to the stage, Dr. Najib.
Thank you. First, I would like to thank the organizing committee for selecting this presentation, which is actually the first time that we will be sharing what is, we believe, a much more powerful and important way of analyzing genome sequences for pathogens and infection control. It is a prototype. It isn't quite perfect yet. It will be better, but hopefully you will see that it is already making substantial progress. So first, disclosures. This work is entirely funded by Genpax, and we're a new company founded in 2020. We really started building our bioinformatics team only in the last half of last year. But we are focused on key healthcare infection pathogens, healthcare associated food and public health. So everyone here is probably familiar with the promoted impact of WGS, and our view on this is very similar to that from the first presentation. It needs to be democratized, it needs to be brought to its full use and potential. But at the same time as people are presenting it, they are raising the issues of the obstacles consistently. A lack of informatics infrastructure, a lack of computing infrastructure, a lack of bioinformaticians. And if the problems for that are difficult in the UK and the US and developed environments, they are even worse in low middle income countries. And of course, the challenge of infection is a global connected one, not just one for the rich and the well-resourced. And we talk about using a bunch of very standard methodologies, but we are not so good at addressing the ways in which they are perhaps suboptimal, and that there are problems within them that are either not recognized or accepted as inherent to them and perhaps can't be solved, or perhaps are impractical to solve. Whereas really, I think these challenges need to be directly named and addressed, because otherwise we can't solve them. So with sequence typing, as was illustrated in its best form, its most sensitive form of WG, MLST in the second presentation this morning, it does achieve scalability for typing and identification by reducing the information to allelic versions of a subset of the information. At its lowest sensitivity, as described earlier, that can be typically 5 to 7 genes. In core genome MLST, it can be typically 50 to 60% of the coding sequences. In whole genome MLST, as was described in the second presentation, it can be up to 20% more, but it is the clearly distinguishable, only one paralogous family member representative coding sequences, which are common to the set of genomes that are compared. So it doesn't address the intergenic sequence. It doesn't actually address all of the coding sequences. And it is doing this in order to make the downstream analysis and identification fully scalable in a kind of tabular bioinformatic fashion, so you can find what you need. And it has modifiable thresholds for what you include or exclude from a final grouping to allow for errors in sequencing and alignment and parts identification. So typically these things range from less than 1% with MLST to around 60% with core and slightly higher with whole genome sequence typing. It is great for what it was demonstrated for, which is the investigation of suspected outbreaks where you already have prior knowledge and you are confirming or denying or pursuing this pre-informed, epidemiologically aware investigation. What it is nowhere near as good for is the investigation proactively of very large numbers of samples where even unrelated strains may have identical sequence types. And this happens a lot, and it's a particular problem in recent clonal expansion species such as Staph aureus and MRSA, which is only 60 years old for the oldest of them, which is in no time in microbial evolution. At the other end, there's whole genome SNP analysis. This is the most sensitive methodology, but it also has significant issues. It has significant issues of sequence error. It has significant issues of selecting or having to select an optimal template genome for the alignment, which can work across the whole of what you're looking at. And as you increase the number of strains and the diversity of strains which are analyzed using it, the actual amount of the compared genome falls and its sensitivity falls. And sometimes this can be quite severe. So for example, a recent paper from the groups in Australia showed that if you are analyzing E. coli and you happen to have chosen a particularly inappropriate template, it can fall to 50% of the genome. And although it's called whole genome SNP analysis, actually it might be better described as common genome SNP analysis, because actually what it analyzes is the comparison of what remains common and conserved and sufficiently specifically comparable across the whole collection. So again, it does not scale well. So it's again very good for the investigation of well-informed prior knowledge, previously suspected clusters, but it's not good for discovery, for identifying the outbreaks that you didn't know you had, for proactive surveillance and detection, and thus a different, more timely, more impactful infection control solution. So we're trying to deal with this. The conceptual features of our new analysis strategy are based on three very simple principles. Accuracy. You must achieve maximum optimal accuracy in order for the noise not to exceed the signal of what you are investigating, especially to do it proactively and at scale. We need resolution. We need to address the maximally accurately addressable genome sequence across the whole of the sample set that we're doing. And typically, we believe the optimum target, which is available, coding and non-coding, non-repetitive, is between 80 and 95% of the total chromosomal sequence space, substantially more than is currently addressed. And if it's to achieve its potential locally, nationally, internationally, across small samples, large samples, and for proactive investigation, it must be scalable. Every component of the process must be scalable, both in terms of information and in terms of computational deliverability, because almost all current methods end up with probabilistic solutions to address the noise problems, such that they are what's mathematically computationally called an NP-hard problem. In other words, eventually you need security service underground computers and entire power stations to deliver the information a week late at too much scale. It must be scalable. So, study we've reanalyzed in this kind of study. It's a study, it's well published, it's been reanalyzed once before. There's 685 MRSA isolates from 610 patients across St. Thomas's, King's Hospital, and an associated community hospital in London. It was done with Illumina sequencing, 150 base pair paired end reads. There's good coverage for most samples within this data set, which is one of the reasons we selected it. And the sequence information is publicly deposited. So if anyone wishes to go back and do analyses or interrogate this work, the information is all there. Of these sequences, 679 was sufficient for quality and coverage to be included in the analysis. And one small caveat, there are some limitations in what is currently available in the metadata. So we don't actually have patient identification information for everything that's analyzed. So where we don't have that information, we have assumed that the patients are from different patients, which in more than 90% of cases is going to be correct. So it's not going to change the conclusions of what I'm going to show, but that's why there's a little bit of uncertainty when I describe missing patients within the large patient cluster a little later. So first of all, scalability and diversity. This is a purely SNP-based, no sequence type reference genome. All clonal complexes sampled result in which the pairwise distance of every one of these strains is independently determined and available and mapped onto this dendrogram. All of the major MRSA sequence types are present, and it was done in a fully automated typing comparison, a comparison method. So you can get the detail of this dendrogram at this high structural approach all the way down to the detail of the branchings in a single dendrogram. It doesn't matter that some of them fall outside of the sequence typing schema. You still see exactly where they are in their relationships. It doesn't matter if they get given a different ST within the schema, you still have complete visualization of the relationships between the strains. And you can see their connectedness and their identifiability regardless of the ST and whether it's found. And here's an example from ST22, and here's one from ST5, and they all lie within this single dendrogram. Now, first surprising discovery, and an important one for proactive detection, is that what you find is if you integrate this data, because we can scale it, so we combined it with an additional 2,200 genome sequences from another study, which is contemporaneous from Cambridge. And what you can see, if you color them in, which is what we've done here, so the blue ones are from London and the red ones are from the large Cambridge study, is that the local hospitals, and they're only 60 miles apart. We did not expect this. We know this happens internationally with strain samples, but this is 2 UK hospitals 60 miles apart where there are exchange of patients between them. They have completely different local microbiomes. So the strains which are in and out of these environments, whether if you zoom in, they're part of an outbreak here, or whether they're not parts of outbreaks, they have different microbiomes with different similarities, such that you can't just assume that the nature of distance is the same between your outbreak and the local population and the outbreak and the larger population. We really didn't anticipate this. And you can see the same thing if we zoom in. Here are the sequence types at ST59, and you can see outbreaks. And actually, this one has the patient identifiers, and you can see 561, 561, 561. These are 3 same patient isolates. And you can see the unrelated patients as part of the connected group. And actually, you can use this structure to determine outbreaks from non-outbreaks, because you can actually see the dynamic range, which is occurring in diversification within individual patients and the outbreaks themselves. Again, another outbreak, not an outbreak. And you can also see the unconnected strains and the way in which the microbiomes are separating. So we did an analysis of the distributions, having seen this, of the distance within the sequence types. Now remember, the ST22 problem is 50% of MRSAs that we're seeing. So you really need to tell apart not just STs, and many of them are ST at every type of sequence typing, they're the same. And what you see is this double peak. And what you actually see here, this is the diversity of the outbreak strains. This is the diversity from the local microbiome. And of course, because we can do the million distance calculations almost for this graph. From our dendrogram, we can see this in a way that hasn't been possible before. And then you see the distance to the normal population. So you've got connected strains, you've got the distance to the proximate microbiome, and then you've got this bigger peak over here just appearing to the right. Then we looked at the SNP thresholds, and we found peaks, and we find that we actually find fewer outbreaks than they thought before. Because by removing the noise and being able to separate the local microbiome, we were able to see that there isn't as much inter-hospital transmission, and the infection cases are fewer in the connected groups. But we did find the same outbreak group, the large cluster. We did find the same breakdown of things. We can see and compare, and we find that our optimum analysis is here at 15 SNPs. And again, if you look at the total number of patients, who's included in the clusters, this is the point at which clusters start to merge. So this is maximum discrimination. And you can see maximum sensitivity, 10 to 15. But again, we now pick this 15 because this gives the optimum discrimination coupled with detection. And we're able to see this information. Now we can analyze all of this stuff with the noise removed. And this, I think, from an infection control point of view, is the most important thing I have to show you. On the left, you have their whole genome SNP analysis done with a large amount of manual time, specialist analysis, specialist bioinformaticians, and you can see the structure that they were able to derive and the suggested connections and pathways between them. What you see on the right is the same outbreak analyzed with our methodology. First of all, they missed, it says 2 to 6 because we can't know exactly what the dashes are in our diagram, but the likely answer is they missed 4 patients in their outbreak in the first place because of noise and it moving things out of their SNP detection threshold. And now you can see a much, much greater granularity and probable transmission chains. It informs the infection control team of a much more likely and more detailed set of connections between the patients and cases so that they can proactively go in there and focus on the right people, not have missing cases, and have a starting model for how they will investigate the routes of transmission, the point sources, source attribution, and the chain of events, which will allow them to have actionable information. And the point is, this can be done now proactively. This isn't the necessary investigation of previously suspected outbreaks. And it also shows that another advantage of doing it proactively is you can get the information to be able to do optimal analysis of your outbreak strains from this local microbiome from the more distant strains. It's transformative. And we have 101 informative SNPs in this analysis. So let's go to that point. As I described earlier, the information that you can access as you do these things at scale normally falls as you add additional strains and additional diversity. Not with this. Okay, we had 101 informative SNPs for what the cluster analysis I've just shown you. If you take the London study as a whole, in their analysis, done at length with a large team, they found 68,997 informative SNPs that they could use for their dendrogram structuring and strain identification. By comparison, we have 142,322, more than double this starting information from which to work, from exactly the same sequence files. And actually, this is pretty stringently filtered at the moment because I want to make sure that I'm presenting the correct answers. This number is probably going to go up as we move from the current prototype. And if you go to the 2,907 sequences, including the Cambridge strains as well, again, the informative SNP information actually goes up because, of course, we're sampling greater diversity. Right? It's exactly the opposite to the established problem of WG SNPs falling information. So, in conclusion, we have a minimally reducing SNP level, multi-scale, direct to identity, don't need sequence typing, analysis. It can address more genome content than any of the alternatives. We've shown a more than doubling of the underlying information that can be exploited from exactly the same sequence files. We can do much broader comparisons, and we've identified this issue of local healthcare microbiomes, which is a fascinating subject, which we will be investigating in much more detail going forwards. We've identified situations where infection transmission has been overestimated with too many clusters. Equally, at the same time, we've identified situations where strains and patients have been missed out from the clusters that have been pursued. The greater information can be used to structure clusters to insist and improve outbreak investigation by infection control. And that's what this is for. That's why we're doing it. And this each against all capability at scale means that this can be applied both to surveillance and outbreak detection, and can be done within your hospital, across a healthcare network, nationally, internationally. That's what it's designed to do. So thank you very much for your attention, and I'll be very happy to take any questions.
My question is, and sort of getting the public vaccines